AI drug discovery is hot, but billions of dollars later, no drug on the market has ever been discovered using AI. Why? Recursion Pharmaceuticals is one of the hottest AI drug discovery companies, especially after NVIDIA's $50 million strategic investment into them.
Perfect. Shares of biotech company Recursion soared 78% today after announcing a $50 million investment from NVIDIA.
We spoke to Imran Haque, SVP at Recursion, to find out what makes AI drug discovery so difficult. So Recursion is one of the hottest AI drug discovery companies at the moment. And I wanted to just ask you, what's the big idea? Like, if everything goes right with Recursion, or what your vision is of the future, what does the world of drug discovery look like in that way?
What we want to do is we want to decode biology. We believe that there is an enormous amount of waste that goes into drug discovery today. If you look at how much money it actually takes on average to bring a new drug to market, it has gone up exponentially over time. And we believe that a lot of that is due to our lack of deep understanding about how biology works. There are a lot of hypotheses that we have that are very linear or single-dimensional in nature. There's, one gene or one protein that causes one disease, and perturbing that will fix the problem. And biology just doesn't work that way. And so the approach that we've taken is to say, look, this way of performing this single-dimensional mechanistic modeling of disease is part of the problem. What we need to do is do a better job of understanding biology. And the way to do that is to collect an awful lot of data about biology, about chemistry, pair that with deep computation, and then actually test those predictions. So don't just make a model and then take it out to the clinic, but actually test those predictions in a continuous cycle. So, if everything works, if that really goes, we think that there's a world in which you could actually get drugs for new diseases, new particular patients with particular diseases, a lot faster, exponentially faster than we are seeing the current drug discovery and drug development pipeline do today. And we think that the root of that is the collection of data, the building of models, the cycling of that back through experiment in order to decode biology.
What's harder than it looks about that? Like, it sounds great in theory. You've been going at this for decades now. What's harder than it looks?
Just about everything, as it turns out. Particularly, mathematicians, computer scientists who come into biology, me among them, right? Will come in and say, oh man, this is easy. I'm just going to take this data, I'm going to throw a machine learning model at it, and it's going to be good. You all didn't know what you were doing. And I remember very clearly the first rotation project that I did in my PhD, where the postdoc that I was working with at the time gave me some data sets and said, okay, now you need to figure out which compound is going to interact with these proteins, right? To predict drugs. I had just come off of Angeline's machine learning class. I was like, all right, no problem. I got this. 3 months later, all of my correlation plots looked like somebody had just sandpapered my screen. There was no correlation whatsoever. And here I am sitting thinking I failed the test. I'm not going to get to do a PhD. They're going to kick me out. I'm not going to make it in this lab for sure. And, I walked into the room to talk about it with a postdoc and with my advisor. And I showed them the results and they said, and thus you're enlightened. Right? So the challenge is, really manifold, right? Even when we go to the smallest scale, when we look at individual molecules and individual targets, there's an awful lot that we don't understand about how compounds interact with particular proteins, how the proteins might change in different contexts and so on. You zoom out, every drug interacts with not just one target, but many, maybe hundreds of different targets in the body at different concentrations in different tissues. We have a fairly poor understanding about how all of those things add up. And as you keep layering on more levels of complexity, what you realize is that the simple reductive models that we have at the base saying, well, this drug hits this target causes this disease, are fundamentally wrong, and that you have to zoom out from that, right? The answer is all of it is hard. Biology is a system that has evolved over billions of years to work. It didn't evolve with a design principle to say, well, this has to be simple and has to be modelable in the easiest way that you'd like. It's more like if you had a billion years of somebody saying, yeah, that's good enough, that'll do. And that's a system that we're trying to work.
I get what you're saying, but then in another sense, when you think of what is possible with a computer chip or what we're able to simulate whole societies were able to play games like The Sims or Grand Theft Auto and do that impressively well. And then I guess on the other spectrum, we're still not able to accurately predict the weather going very far forward. And that brings in things like chaos theory. On a scale of, on one side, you've got Sims and a computer game, and that's apparently quite easy to simulate or model. And then on the other side, you've got this like chaotic weather patterns that we still struggle with. Where do you see the biology and chemistry of drug discovery sitting there? Is it like even harder than, do you think, weather systems, or is it somewhere in the middle? What do you think?
Yeah, if you've got the Sims at 1 and you have the weather at 10, I'd say, trying to do small molecule drug discovery is somewhere out of like, 50 or 100, right? But I think it's a really interesting example, right? So, even over the last 10, 20 years, weather prediction has gotten dramatically more accurate, right? I mean, it was sort of unthinkable that you could just, pull up your phone and have a reasonably accurate hour-by-hour forecast of how things were going to go, or that the 10-day forecast was anything more than pure guesswork. And now they're actually pretty decent, right? Part of that is that, we do have some understanding of the physics that go into these systems, and that's gotten better. A big part of it is that our access to data about these has gotten massively better. So if you look at what's happened in weather, there's a lot more satellite data collection out there. You can actually see what's happening with cloud systems, where they're moving. You can get measurements of the wind and of temperatures all across the globe. And that data then feeds into computational models. At the same time, we have wide networks of weather sensors, temperature, wind, precipitation, and so on, that can then be fed back in to optimize those models. I think those closed loops, like that combination of closed loops of modeling and really high-dimensional, high-throughput data collection, are things that have been absent in biology. And I think that's, it's a great example because I think it's very well aligned with how we see the direction of how to move biology.
I want to give a really basic understanding, like a really simple outlay of what I think Recursion does, and maybe you can just correct me. And that is that you essentially run a load of wet lab studies. So you go and collect this data to try and understand how these drugs could work. And then you develop models using that. And then you apply those models to different therapeutic areas that you think are interesting because there's some clinical need there. Now, I'm sure there's a load of incorrect bits there. Like, why don't you correct me?
So that's actually not incorrect. What I'd say is that it's very broad, right? I mean, that's a process that you could use to describe almost any pharma, where you're going to say, okay, there's a disease, there are systems that I can set up, there are experiments that I can do. I can try to decide which programs might be interesting for a portfolio, and I'm going to advance them that way. So I think the interesting part is actually in the details here, right? So, and what I'll do is I'll establish this a little bit by contrast. So if you were at a typical target-driven pharma company, what you might say is, look, there's this awesome paper that just came out in Cell a couple of years ago, and they think that this gene may be linked to this particular disease. Let's set up an experimental system around that gene, screen a bunch of compounds there, have some chemists work really hard for a couple of years, optimizing that compound, that experiment, put it through some mice, et cetera, and we keep going down. The challenge with that, obviously, I mean, even as I say it, it sounds like a long, slow process. The deeper challenge, in my opinion, is that when you do that, you collect data in the course of these experiments, but you collect very narrow slices or very narrow verticals of data where you have this one target that you looked at, you have all of your compounds tested against that, and then little islands of molecules and related molecules around those. The way that we're going about things is actually to turn that picture around on its head. What if we could collect all of that data up front and then look inside that data to identify the most promising areas up front, and to do so without reference to somebody's favorite Nature paper, right? To try to take a broader look at what we're seeing in known scientific literature, what we're seeing in patient genetic and omic data, and so on. And so to give you an example, the way that we build what we call our map of biology is to take human cells in culture. So these are typically primary cells, sometimes they're cell lines. We grow them in plates that are about the size of your hand, with over 1,500 wells or little spaces for cells in them. And then we'll actually perturb each of those wells with different agents. When I say perturb, that might mean that we knock out a particular gene. It might mean that we put a particular chemical or starting point for a drug on there at a particular concentration. We might put proteins, antibodies, toxins, cytokines on those. And then literally we look at them. We take those cells and we put them under a microscope. And the remarkable thing about biology is that form follows function, in the sense that when the cells are in a different state, when they're doing something different, they actually look different under the microscope as well. And so what we can do is we can take pictures under a microscope of those perturbed cells. And then with the deep learning, machine learning image processing models that we've built in-house, we can relate those different states to each other. The really remarkable thing is that what comes out of these machine learning models is what we call a biologically meaningful space, meaning that directions and magnitudes in the space mean something. Imagine a ball or a sphere, and cells that were unperturbed sort of sat in the center. Maybe you knock out a particular receptor, and that sits over here at x equals 1. As you take wells where you've dosed an antagonist for that receptor, something that blocks that receptor, you'll see the features coming out of the machine learning sequentially move closer and closer to that knockout until you've inhibited it. And in fact, if you dose a receptor agonist, something that activates the receptor rather than inhibiting it, you'll see those embeddings move in the opposite direction. So when we say that we're building a map of biology, in one sense, this is very literal. We're literally taking biology and trying to find out these different perturbations, these different states. Where do they live in space, and how can we get from one to another? What's really neat about that is that once you can do that, you can relate all of these different perturbations to each other. So when we screen a particular compound, a particular chemical on our platform, it's not like in that earlier case where you just get the result for a one target that you screened it against. You can in fact see its relationships against all of the genetic knockouts that you've done in one go. You also get the results of similarity to all of the compounds that you've ever screened in one go. And so every time we run a new experiment, we're increasing the number of relationships, the amount of understanding we have at a superlinear pace. So when we do that, then the discovery strategy becomes totally inverted. Rather than saying, okay, I know I have one gene that's interesting that I found from a paper, let me go build a whole experimental platform and discovery strategy around that. You can say, let me look at the totality of what I've collected. At this point in Recursion's history, about 3 trillion relationships, I think, and ask, what are the most strong relationships? Which ones look like they're real and not just noise? Which ones look novel? They're not things that people have well understood that they've really probed. And which ones look therapeutically relevant? Which ones look like these could actually make a difference in a disease for particular patients? And then you can choose to go after those. So one piece of language that we use about our approach is that we're broadening the funnel at top to narrow it at the bottom. Rather than saying we're going to have a bunch of things and sequentially winnow them down 50%, 50%, 50% until you get down to a drug at the top, we want to look at an enormously large number of hypotheses at front with real data, not just guesswork, and then immediately sub-select those to just the ones that we think are going to go through.
One of the things I find really fascinating about your approach is that you don't rely on the foundational knowledge that we've built up as humans. And that means that by testing so many different hypotheses using the methods you've described, that you might come out with some really left field solutions to different therapeutic issues, right? Or different diseases, because you're thinking in a way, or your machines are thinking in a way that humans have never thought of potentially, right? You're not relying again on this scientist did this, and the next decade this scientist did this, and you might actually come out with some really, really interesting stuff that no one's ever thought of, which is, I guess, one of the promises of these machine learning methods, right?
That's exactly the goal, right? Is to say that, it's really coming from a standpoint of acknowledging that we don't understand everything in biology. There are pieces that we know, but we really don't know everything.
So can I ask a quick follow-on question there? So it sounds like you have like multiple levers to advance the power of your approach. So one lever might be to expand the number of cell lines that you can test on. Another lever might be to expand the chemical space that you can have as inputs into your wells and to observe their effects. A third one might be the power of your machine learning models, and so on and so forth. Let's say you have like half a dozen to a dozen levers. Is there one or two of those levers that you expect are going to drive like the biggest breakthrough in the power of the approach? And I'd be interested to know like how, what are you looking for to as a signal that the approach is working, it's getting better, it's becoming more powerful? Is it the type of insight that you just described, or is it something else?
Well, it's a couple of things. I mean, I think seeing examples like the CDK12 RBM39 example that I've shared are certainly pieces of interest and pieces of validation for us, right? And we've done a few things like that in other contexts as well. I think seeing these progress through the pipeline, right? Seeing some of them like really have success is very interesting to us. Like a number of other companies, COVID was an opportunity for us to really accelerate that process of discovery, and we got some interesting signal there. So, at the beginning of March, April of 2020, COVID was this hugely exploding phenomenon. A lot of interest in identifying if there were drugs or treatments that could be repurposed to actually treat COVID. And so we initiated 2 screening programs. One actually looking at infection with SARS-CoV-2, the causative agent, to see if there were drugs that could help in that sort of like acute phase of infection, as well as one looking at a model of severe COVID, so the cytokine storm that results. And the really cool thing that came out of that was, we did those screens, we published those results. Others went ahead and did clinical trials on those. And when we look retrospectively at the large trials that others did, we got 8 out of 9 of the results right. And we sort of know the reason why that last one didn't go. And so that gave us real belief that this
Wait,
platform...
Imran, sorry, can I butt in?
Yeah.
So
Yep.
I saw this, and it was, you're absolutely right. So it was 8 out of 9. And then the one that Recursion didn't predict correctly, that was dexamethasone, wasn't it? That turned out to be
That's correct.
one of the most effective COVID treatments. Like, what was your take on that? Why did that not? Why was that not predicted?
So the this obviously this is hypothesis, right? Where I haven't gone and done the exact experiments to follow it up. But as I mentioned, the way that we built that screen was we had cells which were perturbed by a cocktail of cytokines, modeling the cytokines that you see in severe COVID cases. Dexamethasone is a steroid. It's going to have anti-inflammatory effects. It was probably acting on a different cell type to suppress the release of those cytokines, right? So dexamethasone would have acted upstream, but we basically short-circuited the way that it would have gotten in there. And so that's not something that would have been modeled in the particular experimental system that we used.
Sorry, can I just so I understand? So you have these human cells which you're experimenting on, and when you say perturbed, you mean almost, and when you're talking about cytokine storm, you're talking about in severe COVID-19 disease, you get this kind of massive inflammatory response by the body, and that goes on to affect the human cells in a bad way. And you were modeling this, is that correct? And then maybe dexamethasone was acting a bit more upstream from where you were looking at. Am I understanding that correctly?
That's exactly right. And so of course the human system is more complicated than this, right?
Of course.
Well, let's assume sort of a 2-component system where we have cells, in the blood vessels or in the lung, right? And you have immune cells, right? Those immune cells are going to be the secretors of the cytokines that are then going to have systemic effects on the rest of the body. The way that we modeled this was by taking just the cytokine mixture and putting them directly on cells, right? It's likely that dexamethasone was not acting there in blocking the response of those cells to cytokines. It's likely that it was acting upstream, preventing cytokine release at all.
Release in the first place, yeah.
That's right.
Okay.
That's right.
I guess that could also be an example of the type of paradigm that you're trying to avoid in terms of the narrow slice of biology and the experimental constructs in the traditional approach that you described earlier on. And would you say that that's just an artifact of a kind of accelerated experimental process in the context of COVID, where a very specific experiment was designed in short order to examine this? Or I guess what I'm wondering is, how do you systematically avoid that type of miss in your broader endeavors beyond COVID-19 and so on, and the other indications you're pursuing?
Yeah, that's a great question. I think it gets to some of those levers that you mentioned earlier, right? So absolutely, at that time, April of 2020, we were, seeing what was going on around the world and trying to figure out what is the most reasonable translational model that we think that we could develop under the constraints that we had available, right? I do think that, in general, there are always going to be trade-offs that you would make with models, right? You have to decide which parts of the system you're going to model and which ones you aren't. At the end of the day, a culture of human cells in a plate is not the same thing as an entire human, right? So, there are things that we do, along those levers to try to minimize or try to reduce like the occurrence of that, right? We can model multiple cell types. We can look at multiple different readouts, whether they are imaging-based, transcriptomic-based. Of course, things, end up going through downstream tests in animal models and so on. One thing that we've sort of optimized our discovery strategy towards is not worrying quite as much about those false negatives, where there is something that in principle like would be good, but you don't get, because the principle is, how do we do this for scale? How can we find enough really good things that if you've got 100 things that work, but you missed one that would have worked, that's still okay. You still come out way ahead.
Or even that you find, I presume, given the value of new medications, even if you find one or two that actually work, even if you were to miss 50 or 100 that don't work, to turn those numbers on their head, it would probably still be a worthwhile endeavor.
That's exactly right. It was something that was that was mind blowing to me. When I was a PhD student, and is still true today, right? I remember my advisor telling me, if you have a 98% error rate on your model, then what you have is like a revolutionary invention, because you're twice as good as the rest of the world, right? Now, depending on where we take the slice, that number changes, but I think the message is still true. Even if you're wrong only 80% of the time, given 90% failure rates, depending on where you look in the pipeline, like that is a revolutionary change in getting drugs to patients.
Imran, can I just build on that? So just to think about like the kind of systematic problem of drug discovery in terms of the cost in industry. So my understanding is that most of that cost is driven by the drugs that people think will work, that go into the clinic, that incur the costs of an expensive trial, financial cost, but also the kind of human cost, and then ultimately fail. In terms of the biggest hurdles for Recursion to overcome to reduce that type of failure at that stage of the process, what are those biggest hurdles, and how optimistic are you that Recursion is going to crack that problem?
Yeah, I'm quite optimistic, right? I think at the end of the day, the reason, well, I certainly hope so, right?
We can't say you haven't put your money where your mouth is.
I mean, it's money and it's time, right? And that's the one resource that's non-negotiable for all of us. So ultimately, the reason why things fail in the clinic, most of the time, it's not that your chemistry is no good, right? It's not usually that you have a molecule that like literally, gets metabolized and excreted too fast, or doesn't interact with the target that you thought it was going to that was going to interact with. We're fairly good at those things, right? Things like metabolism and excretion failures, like those get filtered out in phase 1, if not even earlier, right? And by the time something is getting to those later phases, you have chemical matter. The problem is that you didn't understand well enough the biology with which it was going to interact in order to produce an effect, right? And that's really why we've focused our strategy internally on that biology aspect. Now, we've recently, made acquisitions, we've made investments to bolster ourselves on the chemistry and chemistry machine learning side of things, because those are essential. But we think the transformative piece is really that deeper understanding of biology, which, we've pushed, sort of inside out, right? Starting with the in vitro modeling, and new areas that we're focusing on are deeper connectivity to things like patient and population scale data sets and genetics, in order to drive tighter connectivity with, what are the patient populations that will be responsive for this? For whom is this a good drug? Not just is it a good drug, period.
So just bringing that question back down to those levers or those axes in which Recursion can sort of like continue to develop and build its strengths. Where do you think the greatest impact is going to come from in terms of the acceleration in progress?
What do you mean by the greatest impact?
Let's say there are a spectrum of capabilities that you guys have today, and some of them it would be great to also touch on, if we could, the recent acquisitions, because I see that those have kind of added a few more tools to the utility belt. I guess what I'm wondering is sort of what's the biggest hurdle to progress in the field? Is it one specific aspect across that spectrum of capabilities? Like, is it compute, or the power of the models, or is it like the quality of the imaging data, or is it like the size of the chemical design space, or is it like going from maybe cell lines that are maybe one type of cell in a culture to something like an organoid or some more sophisticated model? Where do you see like the greatest potential progress across that landscape? It could be other things that I haven't mentioned.
Musty was asking earlier in the call, so what is it that's hard about biology? It seems like he should have asked the other Imran, because I think he gave a pretty good summary of the answer right there, right? So the answer is that all of those things are hard, right? We've made our investments in very particular areas, right? To try to say that we think that assessing high dimensional biology in this way is going to unlock what we can do, and that at least for us, we think that given that where we are, then next investing in chemistry is the right move in order to accelerate the progress of that chemical matter. If we're talking about the field as a whole, I think all of those things are potentially true, right? From the machine learning perspective, and this is sort of like, more personal reasons why I came to Recursion. I think the biggest drag on being able to do effective machine learning in biology and in chemistry has been not just access to high quality data, high quality, high dimensional data, but the ability to generate more of it. What you find is a lot of sort of high powered computational teams who don't have the ability to make more data, high powered experimental teams who don't necessarily have the ability to model it well. I think building both the technical capability and the culture to bring those two things together is really transformative.
Imran, can I ask a question with a meta question attached? So this will really challenge your mental RAM, but I think you've got loads of it, so you should be good. When you think about the past decade or the multiple decades you've been in the AI drug discovery space, and then you think about some of the results, and from what I understand, there's no drug that's used in patients currently that's come or been discovered through AI drug discovery. So the question, the actual question I want to ask is, if you had to think of timelines or how optimistic you are about the future, is this like a self-driving cars thing where it's, perpetually 5 years around the corner? Or is this something that we've hit like an inflection point on? And then the meta question attached to that is, do you get pissed off when people like me, I'm sat in my bedroom here. You've been grinding for decades on this problem, and I'm just like, oh, well, there's no drug being used in patients. It's not working. And then you're just like, I'm grinding so hard, you don't understand the half of it. Does that ever piss you off a little bit?
I'll answer the meta question first, and the answer is no, not really, right? It's interesting work and it's noble work, and I didn't get into it because I thought it was easy, right? I got into it because I thought it was a good thing to do, and I'm also sanguine about it, right? Not everything that we try is going to work, but I do have an underlying conviction that there are things in this space that will work, right? And I think that there are a number of things that have to happen here, and that we're also in looking at where the goalposts are. This is a fairly slow feedback system, right? Because if you make a change at the beginning of a discovery pipeline, just the current lag in how long it takes to go from discovery to development means that you've got, probably a decade lag, right? From any change that you see to actually seeing that manifest, on the market, or, a little bit faster than that in trials. And so, this is something where I do think some patience is required, and it's a little bit difficult sometimes to see exactly where that lands. So, coming back to the, non-meta question, right? That's a challenging question. And actually for the reasons that I think the race to claim AI discovered or AI designed drug is so hot, it's hard to tell exactly what that means. And I think different people mean different things by it. Right. That said, I don't really have any doubt in my mind that we will see that, depending on how you think about it, that either we already have seen or we will see drugs in which AI and machine learning methods were essential components to actually designing them, getting them into the clinic, in the future, and not even necessarily in the far future, potentially has already happened, right? There's this joke in the field, right? That like, as soon as something starts working, you stop calling it AI, you call it something else. And so, under that standard, well, you're never going to have an AI-designed drug because AI is just the stuff that doesn't work, right? But, I think the underlying methods themselves are sound. I think the underlying ways of thinking are sound. I think they are necessary transformations in the way that we work and the way that we deal with the complexity of biology and the data involved. And I think the impact there is going to be is going to be undoubtable.
Yeah, just to add to that, like I think there was one drug that was discovered to have a potential impact during COVID, which was baricitinib, which was by a British company, Benevolent AI. I think they published something on this. I think it was actually used in patients in India, and there was a research trial as well to show some benefit there. So the drug was, as you say, I think it's a really great point, Imran, around what does it actually mean to be an AI-discovered drug in patients? Is it one where a connection for an already approved drug has been identified, or one in which, say, the clinical trial has been accelerated using analytics in some way, or one where the fundamental, interactions between a small molecule and a protein is actually, originated by an AI model?
I suppose you could say that AI is in the eye of the beholder, right? Different people would call different aspects of that, and I think that's what makes the discourse in this so difficult, right? Is that different people mean different things when they say AI discovered or AI accelerated.
Right.
And from my perspective, at least, the interesting question is a little bit less the label that you choose to associate with any particular thing, and more, can we zoom out? Like, is there a broader trend of like these methods, these ways of looking at, interpreting, acquiring data, and them making impact on how we do this business?
What are you most excited about, say, over the next 5 to 10 year horizon in this space?
That is a challenging question. 5 to 10 years is an awfully long time in this space, right? As I look at what's happening, I see the convergence of a few things, right? And I realize I sound like a little bit of a broken record here, but I think it's I sound like this because it's real. What I see is an explosion in the flexibility and like comprehensiveness of our computational modeling capacities in biology, paired with the scale and scope of data sets that we are acquiring. I think those take multiple forms, right? Some of them take the form of the kinds of data sets that Recursion is generating, right? High dimensional, high quality data sets about biology where, you can put in whatever perturbation, whatever state you're interested in and try to read that out. Some of those take the form of super large scale observational data sets, right? If I look back 10 years and think about the scale of genetic and genomic data that existed, there was a revolution in our ability to acquire that data with microarrays and then low throughput next generation sequencing, and then the ultra scale stuff we're doing now. We're seeing big changes now, not just in the DNA and RNA levels, but also in the ability to read out things like proteomics, lipids, post-translational modifications on proteins that will just give us a lot greater understanding of what's happening in the biological system. And I think that paired, revolution of data acquisition, both, people and assays, engineered systems to probe at biology, and computational methods to model all of this, that's the thing that gets me really fired up, because that's sort of the trifecta that you need to be able to observe, experiment, model, and close that loop and get better at it.
Awesome. So gather more data, find more and better ways to interact with those biological systems, and then more powerful ways to basically process everything that you're learning.
That's right. I mean, a model is only interesting insofar as its prediction is useful, right? We talk about decoding biology, right? You could read that as understanding biology. And the way that I read understanding is I can make a prediction about what's going to happen if I make a particular change, right? Yeah. And I think our ability to predict the results of an experiment is increasing and will only increase by application of that, right? Do the experiment, see what happened, see if you can predict the next one any better or not, and improve in that cycle.
So throughout your career, have there been any habits or ways you approach problems that have been useful for you? And let me just give you that as the general kind of bait, but then to specify, one thing I really noticed with you, and from talks I've seen you give as well, is that you're able to talk in a way that's very enchanting, to hype people up about what you're talking about and the vision. And I think that's not an easy thing to do, because this is actually it can be quite a dry topic, right? And I think that's something you do very well. So just generally, I'd be really interested in habits and things you've done that have been helpful for you. But then specifically, I'd love you to comment on how you developed this knack for this kind of enchanting / charisma speaking ability.
That's very kind of you. So, I mean, there are actually maybe two different kinds of answers, but in terms of habits, I am sort of aggressively a generalist, right? I think, especially in academia, there's a strong tendency to go down a very narrow rabbit hole, but my interests are extremely broad. One of the things I think has brought me success and understanding is saying, well, look, yeah, I'm interested in the genetics of this and I'll take the biochemistry class, understand the kinetics of how this is going. But I also want to zoom out and see, okay, well, yeah, the fundamentals here are actually the very same thing that we're doing over here. We're just calling it a different name. And this is how we can put these pieces together, right? I think being willing to come out of that rabbit hole, engage with other disciplines, people in other disciplines is really critical to the space, because it is so broad that no one person is going to have all of that in their heads. And being able to understand the words that the folks on the other side of the aisle are using is really valuable. One of the things that I find myself doing a lot at Recursion and elsewhere in my career is simply playing translator, because you'll have different people nominally working on the same problem, nominally using the same words, but the words mean completely different things to each other, right? And being able to bring that together is really critical.
Wait, sorry, can I butt in here? Because I love the point you made about generalism and being aggressively generalistic. But I think one of the fears you can have, especially early in your career, is that by being a generalist, you're kind of on this road to nowhere, and there's a seductiveness in being specific. I mean, I think obviously this book came out called Range, which was very popular, and that promoted being a generalist. But it can be a bit scary, because you're especially in academia, I need to get good at one thing and then maybe I can branch out. And I think, I mean, my question to you would be did you have this fear when you were just exploring your curiosity and doing random things that, oh, my friends have done this and they're going really far into here, and I'm getting this concoction of skills that might not actually be useful? I mean, you're kind of we're studying your winning lottery ticket here, because obviously it's all, it's all turned out to work well, but there could have been a world in which maybe these combination of skills weren't, useful. So do you have any comments on that feeling?
Oh, constantly. I mean, the way that I say it is my computer science friends think I'm a biologist. My biology friends think I'm a computer scientist, and the chemists will definitely not accept me as one of their own. So they absolutely constantly, right? I mean, I think there is a grain of truth in what you're saying, right? In that there's a difference between being a generalist and being a dilettante, right? You don't want to be superficial. To be a good generalist, you actually have to have enough depth in a number of these areas to know what's what, right? But at the same time, it is possible to go so deep on something that there's more gain from learning another thing to do. Now, I think what you're saying is right. It does, not every position is a fit for that, right? And it takes some amount of looking. I mean, before I came to Recursion, one of the things that I explored was actually going back to academia. I kept up my publication record. I was interested in being a professor, and what I realized was that environment really really wanted people who are hyper narrow in one thing. That kind of generalism and bringing together a number of fields, it's changing. I think there are some departments and some disciplines where that is more desired, but there's a strong cultural inertia, for understandable reasons. When you want to push the frontier of a small piece of knowledge, to want somebody who's very narrow. So some of it is a matter of finding the right environment for what it is that you want to do.
And sorry, I cut you off. I don't know if you wanted to finish off on the kind of habits and things that have helped you become such the Tony Robbins of biotech.
I'm not sure. Should I like that? I'm not sure if I like that. Anyway, one thing that I would give a shout out for, and it was something that I did that I think is a little bit unusual for folks who go into the sciences, was I was an avid speech and debate person in high school, right? This is something where, most of my friends from those days are now lawyers, right? Sort of obvious why I would select for that. But the experience of needing to be able to communicate clearly, concisely about complicated topics to a mix of audiences, some people who may be deeply versed in the technical aspects of what you're doing, some of whom may be lay people, is a skill that has really stuck with me and that I found to generalize across all the contexts that I've done. So, I remember having relatives ask, "Oh, but you did all that debate. How come you're not taking the LSAT? Like, isn't that a waste?" Absolutely not a waste. Fundamental skills that have served me well through the rest of my life.